My name is Hannah Sizemore, and I'm going to be presenting my project, Investigation of the Activity of tRNA Modification Enzymes Trm732 and Trm734. So post-transcriptional modifications of tRNA are critical for proper cell function. Trm7 is a type of protein that puts modifications on tRNA. Specifically, it's a methyltransferase, so it adds a methyl group to the 2' OH of ribose molecules. Trm7 interacts with another protein called Trm732 to put a methyl group on residue C32 in yeast tRNA-phi. It also interacts separately with Trm734 to modify residue G34 in yeast tRNA-phi. Previous studies have shown that yeast lacking Trm7 are sick, as well as yeast lacking both Trm732 and Trm734, but single mutants of Trm734 and Trm732 are healthy, indicating that these proteins are important for function, but their exact role is not yet known. The human ortholog of Trm7 is FTSJ1, and mutations in this have been linked to non-syndromic X-linked mental disabilities, among other things. Budding yeast is a good model organism for functional and genetic studies of proteins like FTSJ1 and theta. So we're going to use yeast to specifically try to identify important residues in Trm732 and Trm734 in yeast. First, we did a sequence alignment to determine which residues in the proteins are conserved. From this, with Trm732, we identified RRS750, GLP754, HG976, and RH702 as regions of conservation. In Trm734, we identified SDD200, RLW206, WSH220, and HRL613. So we used these residues that we identified to create variants of these proteins. We replaced these residues with alanine residues via site-directed mutagenesis. There are a couple different ways which we wanted to look at the activity of these variants. First was genetically, so we plated them on selective growth medium and analyzed the growth levels. With this, for Trm732, we found that our RRS, GLP, and our RRS-GLP double variant all had significant growth defects, while the HG and RH variants appeared to grow normally, similar to wild type. With Trm734, we saw that our SDD, RLW, and WSH variants also had growth defects, indicating that these residues are important for function or for growth. And again, we see that HRL613 had normal growth levels, indicating that it is not as important for function. It grew similar to wild type. The second way which we wanted to look at the activity was biochemically. So we used an instrument called the UPLC, which allows us to actually quantify modification levels. And the data that we saw from this was similar to what we saw genetically. RRS GLP had significantly less CM levels, while HG and RH had levels similar to wild type. And with Trm734, we see SDD, RLW, and WSH had decreased GM levels, while HRL had higher levels. The crystal structure for Trm7-Trm734 interaction has been solved, and these residues have been identified as being next to the active site, which is similar to what we see in our data with these residues. They are identified as areas of importance.
Hi, Hannah. Good job on your poster, your presentation. Great job on all your hard work. And I'll miss having you in the lab next year, but on to bigger and better things. Take care.